Mork Family Department Student Research Symposium. I won't talk too long now. I have some more remarks towards the end, but I think we should begin with our first oral presentation. Nazanin, if you're ready to come on up. And so this is a talk from the lab at Crandall. So Nazanin. Thank you.
Really quick to give you a background about our research. Nanoparticles can be found in the liver, blood vessels, and heart and other organs, and have been associated with pathogenesis, clotting disorder, and other health effects following inhalation. Their most likely route of entry into the systematic circulation is across the epithelia of the lung, especially alveolar epithelium, with its large surface area and very thin barrier thickness. Also, newly developed manufactured nanoparticles are being increasingly utilized in industrial applications and biomedicine as cutting-edge tools for diagnostic, imaging, and drug delivery. Due to concerns about the health effect of the ambient particulate, especially the nanoparticles, it is really important to understand how these inhaled nanoparticles gain access into the systematic circulation and the specific mechanism of their uptake. To investigate the effects of nanoparticles on lung, we use in vitro models. Distal lungs consistent of two different cell types, alveolar epithelial type 1 cell and alveolar epithelial type 2 cells. As a matter of number, 60% of the number of the cells is type 2 cells and 40% is type 1 cells. But because of the morphology of type 1 cells, they cover most of the distal lungs, and more than 95% of the surface is covered with type 1 cells. Type 2 cells are known to be involved in synthesizing and secreting surfactant proteins, but type 1 cells' main role is in gas exchange. It's really hard to isolate type 1 cells because of their specific morphology and they are sticks to the membrane, but in vivo, in the case of injury to type 1 cells, type 2 cells transdifferentiate to type 1 cells. We have seen that the same phenomena happen in the cultures, and in cultures, type 2 cells, after being in cultures for 3 to 4 days, they transdifferentiate to type 1 cells, and they show the morphology of type 1 cells. You can see really thin cytoplasmic extension and protruding nucleus. Also, they also express the phenotypic markers of type 1 cells. We use these type 1-like cells in culture to investigate the effect of nanoparticles on them. You can see that they show really high barrier properties, transmolecular resistance of about 2.5 kiloohms square centimeter, which is consistent with the expected barrier properties of the distal lung separating air and blood spaces. Okay. This slide just really quick shows the method we use to look at the effects of nanoparticles on lung. We were able to get polystyrene nanoparticles from molecular probes, which are fluorescence labeled. They come in two different sizes and different surface with different surface characteristics. And we apically expose these cell monolayers to these nanoparticles. And by measuring the accumulations of these nanoparticles in basolateral, we could calculate the transport rates of these particles. This is not a new slide I'm showing, but it's the starting point of this research and was really exciting for us. It's already published in Nanomedicine. What we observed was not only these nanoparticle translocase atrostrat alveolar epithelial cell monolayers, but also positively charged particles transport 30 times faster than negatively charged particles when they have a similar size. As a matter of size issue, the smaller size particles go 3 times faster than bigger size particles when they have a similar surface characteristic, indicating that the trafficking rates of these particles is strongly dependent on their charge and to some degree to their size. One of the main objectives of this research was understanding the mechanisms of transport of these particles across the alveolar epithelial cells. Here in this cartoon, I summarized the different ways that any known nanoparticles or macromolecules can go through any known epithelial barrier, including rat alveolar epithelial cells. It's possible that the nanoparticles can go through the cell-cell junction paracellularly. It's possible that they use any known endocytic pathways for transport, or there's other possibility that these particles somehow dissolve in the cell, in the plasma membrane, or make a so-called hole and go through the cell membrane. For discriminating between paracellular and transcellular pathways, we do the cofocal microscopy. You can see on the first one that these nanoparticles, this is the one that I'm showing, the mid-section cofocal micrograph of these cells exposed to carboxylate modified 100 nanometers PMP, that these nanoparticles are mostly inside the cytoplasm and not on cell-cell junction or nuclei. I have done the Z-stat, and from top of the cells to the bottom, they are still inside the cell. Also, when I expose the cells to EGTA, which is the calcium chelator, and loosen up the junction and reduce the RT to 0 and measure the flux rate of these particles, still the flux rate across the EGTA monolayer treated with EGTA and non-treated was the same. And in cofocal microscopy, still the nanoparticles are inside the cytoplasm, not on cell-cell junction or nucleus, indicating that these nanoparticles most probably go through transcellular pathways but not paracellular pathways. So we go after the endocytosis to see if they use any endocytic pathways for transport. Here this slide summarizes the effect of methylbeta-cytodextrin, the known inhibitors for caveoli-mediated pathways. When you expose these cells to methylbeta-cytodextrin, the flux rate of none of the PMP we used in any size or charge didn't decrease, rather than when we had the positive control of chlorotoxin B, which is known going through caveoli-mediated pathways, and the flux rate of chlorotoxin B decreased significantly. So just think that these particles do not use caveoli-mediated for pathway, for transport. We also look at the colocalization profile of these PMP with caveolin 1 and chlorotoxin B as a markers for caveoli-mediated pathways. You can see in the panel A that here it's shown the positively charged particles and the caveolin 1 markers do not colocalize. In panel A2, I'm showing just intensity profile of these different channels to just emphasize that the green particles do not colocalize with the caveolin 1 in red. Also, when we look at the profile of these particles with chlorotoxin B, that you can see that after 1 hour of exposure, they are in the different levels. And then with the molecules known going through the caveoli-mediated pathways, these nanoparticles do not colocalize, and they don't use the caveoli-mediated pathways. These data were consistent with our pharmacological inhibitors study.